So we're switching gears a little bit, and now we're going to be looking at using a Python library called Biopython to analyze protein structure. Specifically, we're going to be working with a file type called an mmCIF. So an mmCIF file is a newer version of a PDB file, which a PDB file is a file from like the protein data bank that contains usually experimental coordinates of a like protein or nucleic acid. So if we look at, RCSB protein data bank, this page you may be familiar with, we have protein structures in the protein data bank. So for example, here on this, so I'm just at rcsb.org. Here there's 7qpq. So that's an entry in the PDB. That's the PDB ID. And I'm gonna let's just click on this one. That will take us to a page that has information about this, this structure. So this is you can read about what it is here, but basically when something is in the protein data bank, that means the coordinates of the atoms have been determined using some kind of experimental method usually. So here's like an X-ray diffraction, and it gives you information about like the resolution and other experimental parts of the structure determination. But an mmCIF file is a file that gives information about the experimental conditions under which the structure was obtained. And it also contains the coordinates for the structure. And then if there is a journal paper that was published about it, it contains information about the citation for that journal paper as well. So that is what an mmCIF file is. Today, we're going to be using a library called Biopython, and Biopython has functions in it that are related to biology. That's why it's called, or sort of molecular biology, and that's why it's called Biopython. We're going to be using a really small part of the Biopython library. We're going to be using the part of the Biopython library that's used to parse or to read in protein structure files, specifically the mmCIF format. The structure that we're going to be looking at, I'm just going to click here, is 1MBN. And I see a raised hand. Is there a question?
Yeah, I have a question. Does the Biopython package have any functions to analyze like gene expression data?
I don't actually know Biopython well enough to tell you that. Paul, have you used it for that?
I haven't used it for gene expression data. I have used it for protein analysis, and it has a lot of function for protein analysis for like calculating molecular weight, isoelectric and things like that. I haven't looked at gene expression data. So probably your best bet would be to just search it, Biopython gene expression data, and find out what it does.
Right.
Okay, thank you.
So here, so it might do that because Biopython really does a lot. And then there are some other, there are other libraries that you can use as well, but Biopython is commonly used for this kind of application. So I also have biopython.org open. So this is where all of the documentation for Biopython would be. So if you want more information about Biopython, this is a good place to start. So we are going to be analyzing in this notebook this crystal structure called 1MBN. And this is like one of the first crystal structures that was discovered. So this one was deposited in the protein data bank in 1973, and it's the crystal structure for the protein myoglobin. And so we're going to be using Biopython to look at that. So I mentioned that we're specifically going to be parsing mmCIF files. You can download those from the PDB. So here, if I click download files, you'll see like one of the options is PDBX mmCIF. We have included the mmCIF file for this protein in your lesson materials. So I'm going to click over here to my lesson materials. The notebook that we're working in is 04 Biopython. But I'm going to show you the file that we're going to be analyzing. So I'm going to double click in data. I'm going to double click in PDB files. The file is 1mbn.cif. So I'm just going to open that so you can see what it looks like. So this, the mmCIF file is a text file that has a specific format. All of them will look something like this, but the data that is in them will be different. So anything that starts with this underscore is like data that is associated with the protein structure. And these words that are here are defined, or specifically defined, and there are places where you can look up ones that you might expect. So so they're all probably going to have an entry ID, and you'll notice that's 1MBN, that's the protein, that's the PDB ID. Then it has some other information here. So you'll see that it has the initial deposition date. This one was 1973. Here it has information, you'll see it has information about the citation for this protein. So here's like the title. So really, if I keep scrolling, so here's for example, the sequence of the protein. If I keep scrolling, here's the 3-letter sequence. So this is quite a long file. It's got a lot of data in it. And then if we keep scrolling, here is where we'll find the coordinates for the protein. So in part 1 of our workshop, so in the Python Crash Course part 1, we talked about how you can read a file using Python, and we kind of like pulled information from a PDB file ourselves. But actually, you usually won't have to do that because Biopython has functions written in that allows you to access all of this information that is in your mmCIF file. So you don't have to write something yourself to pull that information out. Biopython has a function that does that for you already. So this is where we're going to start using, if you remember in the Python primer, we covered Python dictionaries. So they're going to become really important because Biopython really likes dictionaries because of all of this data that is in the front of, or the beginning of the mmCIF file. Okay, so, I accidentally moved this. I don't actually understand what just happened. Okay, so if you opened your CIF file, you can click back to your IQB Crash Course 2023 and then open 04 Biopython, and it will open this notebook for you. And we are going to use Biopython to read that 1MBN structure that we talked about. Okay, so this goes over basically what we've talked about already. The mmCIF file format defines the 3-dimensional structures and it provides the molecule, ligands, host organism, lots of information about the protein. If you want to read more about the mmCIF file format, you can click this link, and that's going to open a page for you from PDB 101. That's a beginner's guide to PDB structures and this file format. So if you're interested in reading more about that, I encourage you to check out that link. And then here's the information that we already talked about, where we have these entries in the PDB along with their data. So let's see how we can use Biopython to read this, this information. So we are going to start by importing functions that we need from Biopython. And we're going to look at 2 different ways you can use Biopython to analyze an mmCIF file. The first one we're going to look at is going to convert the information in the mmCIF file to a dictionary. So we are going to import something from Biopython called mmCIF to dict. So we'll do from bio.pdb.mmCIF CIF, the number 2, and then DICT. Import mmCIF to DICT. So here we're importing this object or function from Biopython, and this is going to convert an mmCIF file to a Python dictionary. So that's what mmCIF to dict means. And then, so after I have my import written, I'm going to create another variable called PDB info. And I'm going to call it mmCIF to dict. And then in parentheses, we're going to put the name of the file we want to open. And we're just going to type the file name. We're going to do data slash and then PDB files and then 1 mbn.cif in quotes. So the reason that we typed that was because we have a data folder, and in that data folder, we have another folder called PDB files, and that's where the file we want to read is. And then if we want, we can check at the end and print it just to make sure that we got the information out of the file. Okay, and you'll see that this outputs quite a bit of detail, but it's about as long as that mmCIF file was. But what it's done is now we have all of the information from that mmCIF file in a Python variable called PDB called PDB info. Okay, I'm just going to comment this out because I don't think I need to see all of that, and I want you to be able to see the code that I wrote. If this worked for you, can you use your green check mark, please? So we said that this function is going to read everything from the mmCIF file into a Python dictionary. In our Python dictionary, you'll remember in the primer, we said that a dictionary allows us to define key value pairs. So it's basically saying, like for a key, I can look up what the associated value is. In this dictionary that we pulled out from mmCIF to dict, our keys correspond to things like this. So the parts that began with the underscore. So I'll demonstrate. We can do, if we want, for example, to see information about the cell length, we could do PDB underscore info, then square brackets, and then I'm going to type the key name for the information I'd like to see. So I'll type underscore cell dot length A. And then that will print the cell length for me. Okay, if you're interested in seeing all of the possible keys, you can use this function pdbinfo.keys. It's going to print out a lot for you, so I'm not going to execute it, but this is how you can see all of the keys that are in your dictionary. Okay, so now that we have this dictionary, one thing we might want to do is print out all of the info associated with one type of data. So maybe we want to see all of the cell information. So if we want to do that, we want to print all of a particular type of data of interest, we can do that using a for loop. So in the Python primer, we talked about a common way to loop through Python dictionaries. And we discussed using the syntax of dictionary.items. So we're going to use that again. So I'm going to type for key, comma value in PDB info.items, and then do open and close parentheses. And then I'm going to use an if statement. So I'll do if, and then I'll type underscore CELL, and I'm just going to put a dot here in key. So this is saying if the key contains the word cell, and then I will print the key and the value. And that's going to show me all of my cell info for this structure. Okay, so if you are interested in learning more about the keywords that might be in an mmCIF file, I've included a link to a web page here that gives, this is actually from the worldwide PDB. So let's click on that. And this will show you information about all of the categories and groups that could potentially be present in a PDB file. So let me just click, I'll click on one of them. So let's click on cell, or I'll click on cell to show you. And it gives us information about like the cell keyword. And I also think it's interesting, this website will show you approximately the percentage for how many protein structures tend to have this entry. So not every protein structure will have it, but this particular keyword is contained in about 94% of protein structures. And it will have sort of more of an explanation for you here if you're interested in that. Okay. So let's keep modifying this a little bit. Let's look at something else in the in our PDB file. And I think the category that is one category that is really interesting is the entity category. So if you were to go to this website that I linked you to and you looked up what entity is, you'll see this explanation. So it says data items in the entity category record details such as chemical composition, name, and source about the molecular entities that are present in the crystallographic structure. So let's take a look at what the entity category looks like. So I'm going to use the same kind of for loop structure that I used before, and I'm trying to keep as many things on the page as possible, but I think what I'm going to do is I'm going to copy this so that you can see it, and I'll just edit this. So I'm going to add another if statement to my for loop. I'm going to say if, and this time I'm going to use underscore entity, and I'm going to put a dot here in key. And then I'm going to print key and then the value. And that's going to add some additional information to what I can see about my structure. Okay, so I've now added printing information about the entity category to my code. And now I can see information about the entities, or it's really kind of like the molecules that are present in your file. So here it's telling me that my structure has 3 entities, like ID1, ID2, and ID3. It's telling me that the first entity is a polymer, and then the other 2 are non-polymers. So this is basically like either usually a protein or nucleic acid, and these are usually small molecules. Then it also has a description of what those entities are. So the first one is myoglobin, then we have a hydroxide ion, and then we have this protophorin 9 containing Fe or iron. So this is like a heme group, which we'll talk about that in just a minute. Then it has some other information like the molecular weight for each entity that's in the mmCIF structure. Okay, I'm going to clear the reactions. If you are with me, use your green check mark. Okay, so what we're doing so far is we're looking at how we can use Python to pull information out of this mmCIF file. So we saw that we have all of that information in text when we just opened the text file. And now we've used Biopython to get all of that information accessible to us in a dictionary. Okay, so I'm going to add a little bit more to my code. You'll see here that for many of these things we're looking at, there's a question mark. So that means that it actually doesn't have a value. So I'm going to edit my code a little bit more. I'm going to copy this to my clipboard. I'm going to bring it down here. And I'm going to add one more thing to my for loop. And so I'm going to add if. I'm going to say I only want to print this if it doesn't have a question mark. So now I'm going to say if I only want to print these things if I don't have the question mark in value. And then I'm going to have to highlight these other two if statements and put them underneath this. And it's actually not in value. So what we're just doing here is just adding more to our for loop so that we don't see all of these entries with a question mark. And then it's just a little bit cleaner for us to read. Okay, so a little exercise for you in these 3 cells. We'll just pause here for 2 minutes or so, but here are some keywords that I want you to try to use. So I want you to try to figure out what organism this came from. So to get the common name, so that's like to get the common name of the organism that this came from, you use this key. You can also get the scientific name, and then this key will allow us to get the protein sequence in the 1-letter code. So we're going to pause here for 2 minutes and go ahead and try to use Python to get that out of the dictionary. And then we'll wait a little bit, but maybe after a minute, if you have the answer, you can post it to the chat. So go ahead and try to get that information from the dictionary. And if you've finished, use your green check mark. Okay, does anyone want to post what the common name for the organism that they found was? Yes, correct. So how we would do that, we can do PDB info, then we put square brackets, and then we can just, I'm just going to copy this actually. And it's going to print for us what the common name is. If we do the scientific name, we're going to see, so we can also do the scientific name. Oops. So that's the scientific name for sperm whale. And then you can also get here, this is the 1-letter code. So this will give us the sequence for the protein. So we can do PDB info. And so here we have the 1-letter sequence. So we had a question. So I do want to address a question. So are these keys strictly standard in mmCIF files, or some can change from file to file? These are standard key names in mmCIF files, but not every mmCIF file contains every key name. But if you're ever looking for what the common name for an organism is, if the mmCIF file contains it, it will be present under this key. So these are standard, and you can see their definition on the site that I linked earlier. Okay. Then there was another question I'll just address because, well, so someone pointed out that they didn't put the underscore here and it still prints out. That's because it looks for just that the key contains it. So since it contains entity, then it still works. So you didn't necessarily need the underscore here because we're using the in keyword. But if you're using these square brackets, it's going to be really important that you put the underscore there. Okay, so that's how we can get information from the PDB file in a dictionary. But really what's valuable about having an mmCIF file is that we have the 3D coordinates for the structure. So it allows us to do things like look at what parts of the molecule are interacting with one another. So in our analysis above, we saw that this structure, and I also told you that this is a myoglobin structure, but this myoglobin structure contains an entity called this, which I have trouble pronouncing, but I'm just going to call it a heme group from now on. So what this is this is a molecule that has iron in the middle, and it binds to myoglobin, and it also binds oxygen, and it carries or it stores oxygen in your cells. So in myoglobin, the iron atom reversibly binds oxygen to carry oxygen to the muscles. And so what we're going to do now is we're going to use Biopython to analyze how this heme group binds to our myoglobin protein. So later in the workshop, we're going to learn how we can use Python to make a visualization of the protein structure. And this looks better if we zoom out. It was a little fuzzy, but hopefully it looks better for you. But here is a picture of the protein structure that we're analyzing. And I've highlighted here this heme group for you and where it's present. So you'll see that it's bound to our protein. And what we want to do is we want to use Biopython to figure out what the residue name or what amino acid this heme group is binding to our protein with. So in order to do that, we're going to have to use a slightly different way of reading PDB files, or I keep calling them PDB files, but it's an mmCIF file. The mmCIF is really, I think, just an updated PDB file, so I get it wrong sometimes. But we want to use a different function in Biopython so that we can analyze the three-dimensional structure of our protein. So this is a second thing. So we're going to import something else from Biopython. Instead of importing mmCIF to dict, we're now going to import something called mmCIF parser. So I already have this imported here for you, but we're doing from bio.pdb mmCIF parser. We're importing mmCIF parser. So to read a three-dimensional structure using Biopython, we're first going to create a parser, and we could use this on, the reason that we do this is because we could use it on several mmCIF files, but we're just going to use it on one. So we make something called mmCIF parser. And then I'm going to tell it quiet equals true. I'm going to be using this to read in an mmCIF file, and I don't want it to give me like a lot of printed output. So I'm putting quiet equals true here. And then I'm going to actually parse the mmCIF file or create my structure object using a function that is on this parser. So I'll do structure equals, and then I'm going to use this parser object or this parser variable I just made. Now I'll do parser.get structure. And then when you use this function, the first thing that we have to do is give this structure a name. So I'm going to name it myoglobin. And this name could be anything that you wanted it to be. And then we have to put the location of the file that we want to read to make the structure. So we'll put data and then PDB files, and then 1mbn.cif. So just the name of the file that you want to read. So we put the path. So we're looking in our data folder, then in our data folder, we're looking in the PDB files folder, and then the name of our file is 1mbn.cif. So it's the same file we were reading earlier. So I'm just going to execute that. And if everything worked correctly, I should see no output, but I should have a number that appeared here in the cell. So if this worked for you, can you use your green check mark, please? Great.
I have a quick query, Jessica. Why did you write quiet?
Because if you don't write quiet, we can try it without quiet. Oh, it doesn't matter for this one. For other structures that I analyze, sometimes it gives a lot of output if there are some things that aren't, maybe that it doesn't understand in the file. But sometimes for some structures, it will like print a lot of information for you when it's reading it. So I just put
can
quiet.
we write in place equals to true?
Hmm?
Can we write in place equals to true?
You cannot. So in place is something that's special for a pandas data frame.
Oh, okay.
Modifies the data frame in place.
Okay, thank you so much. That was.
Yes. Okay. So if it looks like nothing happens, then that means that it actually worked. And now we have a variable called structure. And if we look at, I could add here type of structure. It's going to tell me that the type of my structure is a Biopython PDB structure. And so this variable has a specific, it has a lot of information associated with it that we can't really see here, but there are different ways that we can use it to get information about all of the atomic coordinates. We can also get information about the residues. So in a protein structure or a nucleic acid structure, a residue will refer to either an amino acid or a nucleic acid base. But we can get information about all of the residues that are in this file. And specifically, it's kind of laid out in a hierarchical way. So this is actually from the Biopython documentation. It uses something called a structure model chain residue atom architecture. But what that means is a structure, which we have a structure here, it consists of different models. So sometimes you'll maybe read something, a protein structure where there are several different coordinate sets. So there are different models. A model consists of chains. So these would be like protein chains, chain A and chain B. A chain consists of residues. So these residues would be the amino acid residues. And then a residue consists of atoms. So that's how Biopython is laid out. That's the way many structural biologists think about proteins, but it's an efficient way to deal with the structure. So whenever we have, I'm telling you this so analysis that we do later makes sense. If we have atoms, we can get the parent, like we can get the residue that that atom belongs to, because since we've used Biopython to read in our structure object kind of knows what residue each atom belongs to. So to remind you of the analysis that we were doing, we want to figure out what amino acid or how this heme group is binding to our protein. So what we're going to do is we're going to loop through the atoms. We're going to find our iron atom, and then we're going to ask Biopython to tell us what the neighbors of that iron atom are. So the function that we'll use, we have some of this written in already. We're going to make a variable called atoms, and we're going to use a function on this structure variable called getAtoms. And then here I've printed the type of atoms, and this is going to be important because this is going to be a Python concept that you've maybe never heard of, but will be really important for understanding the way Biopython works. So here, I'm going to create a variable called atoms, and I'm setting it equal to structure.getAtoms. And then I'm looking at what the data type of atoms is. And so if I execute that, it's going to tell me that my atoms variable is something called a generator. And this is something that we haven't talked about before. We did not include it in the Python primer. And this is not really this is a little bit beyond like basic Python stuff. But Biopython really likes to use this data type called a generator. And you would think, I would have expected this data type to be a list, but Biopython makes a different choice about what it returns to you. So a generator is a collection. So it's going to be a collection of atoms, except that when we use it in a loop, it really only loads one of those atoms at a time. So I can't index into this atoms list, but I can loop through it. And one thing to know about this is that you can actually only loop through a generator one time, and then you would have to recreate the generator again. And this is done usually, I think, because there can be a lot of atoms in a protein structure. So you might just want to load one at a time. So that's just something to know. When we use structure.getAtoms, it's returning to us something called a generator. When we have a generator, we can't access all of the atoms at once. And then we can really only loop through a generator one time. So since I know that, I'm actually going to here create a variable called atom list. And I'm going to convert my atoms to a list instead of a generator, just so I don't have to recreate those atoms again when I want to use them, because we're going to use them more than once. So what we're doing in this for loop, we've set up a for loop. We're looping through atom list, and we want to find our iron atom. So for every atom, I'm going to look at what the element is for that atom. So I'm going to set if atom.element, and then I'm going to use equals equals, and then I'm going to put in quotes FE, capital F, capital E. And one thing to note is that in Biopython, these atom elements all seem to be uppercase for some reason, but we're going to say if the atom that I'm looking at is iron, then I'm going to save that atom. I'm going to create a variable called iron atom and set that equal to the atom. And then I'm going to get the coordinate of that atom. I'll do atom dot get coord. So this is the function in Biopython to get the coordinate. And then I will print what I've gotten from my for loop. Okay, so I'm going to execute that. And then it's going to show me that I have this atom FE. And then these are the coordinates. So the X, Y, and Z coordinate of my iron atom. If this worked for you, can you use your green check mark, please? I'll also show you, well, that works okay. Depending on how I think I set it up correctly, but the generator can sometimes give you problems here. Okay, so we found our iron atom. And now what we want to do is we want to measure the neighbors of our iron atom so we can figure out which residue it's bound to. Should I pause here for a minute? I'll just pause briefly.
We're not seeing a lot of questions in the chat, Jessica.
Okay.
So I think things are going okay.
Okay, I got to slow down on the
Oh,
reactions. So
There,
Um,
there is a question here. Is there a way to get what residue or chain a particular atom is on?
yes, we're going to do that.
Okay,
So if we want to get, so we're going to use a function called get parent soon. So basically you have to have the atom that you want the residue of, but we'll do that as part of our neighbor search. So we want to print the residue that this heme group is binding with. Okay, so we pulled out our iron atom, we found our iron atom, and we have the coordinate. And now we're going to define a cutoff distance that's going to define our neighbors. So we're just going to do a distance search to see which residues are close to this, this atom. So I'm going to define a cutoff distance of 4. So I'm going to create a variable called cutoff distance and set it equal to 4.
Quick question.
And, yes.
Is that by default in angstroms?
Yes, so it's going to correspond to like whatever the units are for the mmCIF file. And for
Okay.
mmCIF file, it's usually going to be angstroms, but Python itself doesn't actually know what the units are. So if I put 4 here, it's just going to assume 4 units, the same as whatever mmCIF file it read in.
Okay. Is there a way to like, or so you would have to like look at the CIF file to see what the unit is?
Yes,
Right.
I imagine that they're all angstroms, though, but
Okay.
you'd probably check and see if there's a unit keyword.
Okay.
Yeah. But anyone can write an mmCIF file, so I guess there's no guarantee. Okay, so we defined that cutoff distance. We're going to set it equal to 4 angstroms. Next, you'll see I added an import here for a function in Biopython called neighbor search. That's going to let us search for neighbors for whatever atom we're interested in. So we now will create a variable called neighbor search. And we're going to set that equal to. So notice here my variable is named all lowercase with the underscore in between. We're going to set it equal to neighbor search. Has to match this import that we did exactly. And then we're going to put in the atoms that we want to use for our neighbor search. So I'll do atom list. So for this neighbor search, we're going to be searching for neighbors in all of the atoms, but you can imagine that if there was only a subset of atoms you wanted to look for neighbors in, you would change what you put in this neighbor search. And then we're going to actually perform the search. So I'll have a variable called neighbors, and I'll set that equal to neighbor underscore search. So the same variable that I defined here. And then we'll do dot search. And then I give it the coordinate. So I'll give it iron cord, the coordinate that I got up here. So I give it where I want to look, the coordinate of what I want to find the neighbors for. And then I give it the cutoff distance that I defined. So I said my cutoff distance was 4 angstroms. And then I'm going to print the number of neighbors here, and I'll use my formatted printing or my formatted string, and I'll type the iron atom has, and then I can do length neighbors. Oh, what did I mess up here? I think, so if that happens to you, I think that's what I was remembering having problems with. It has to do with the generator, and I didn't quite divide these up very well. But if that happens to you, go back up here and execute this cell again, and then re-execute till here. So you should get that the iron atom has 21 neighbors. Okay, if that worked for you, use your green check mark. Okay. Okay, so now we're going to continue our analysis and get the residue that these neighbors belong to. So I have this for loop set up already. We're going to loop through the neighbors, and this is where the hierarchical part comes in. So for a neighbor is an atom. We can use get parent to get the residue that this atom belongs to. So we can create a variable called residue and set that equal to neighbor get parent. And then here we're printing the res name, the residue name for that residue, and then also the residue ID. So the residue has a name, and that's the name of the amino acid it's part of if it's in the protein. And it also has an ID, and that's associated like with the mmCIF file. So if I print that, I'm going to see that I have histidine, and then here's some information about the ID. The important number here kind of is the one in the middle, 93. So that is the ID number. So we can see that this is histidine 93. And then these HEM, those are actually the heme group that are in the same molecule as the iron atom. So the lines that say heme are atoms that belong to the heme ligand. So we're interested in how the ligand binds to the protein. So we're going to modify this for loop a little bit so that we're only printing the residues that don't belong to the heme group.
Okay. Hey, Jessica, could you back a scroll up a little? I'm just having, I don't know, with having the, when I get the error about, in the previous trend, it said tuple index out of range.
Yeah, so start here and run that again. It happens because it was a generator and we're executing this line should really be up here. I should have put this line up here, but I put it in an inconvenient cell. So just start here and run your code again.
Okay.
Yeah, it has to do with it being a generator, and you can only use a generator one time. Okay.
Let me see if I catch them. Thank you.
Okay. Right, so we've printed the residue neighbors. Here we're seeing that it's printing information about this heme group. We just want information about the histidine. So I'm going to modify this for loop a little bit. So now I'm doing for neighbor in neighbors. This is very similar to before. We're getting the residue. So I've added here that we're getting the parent of the iron atom. So that gives us the residue name for the iron. And now in our for loop, we're only going to print the neighbor information if it doesn't belong to the iron residue. So if the current residue that we're looking at is not equal to iron residue, then we're going to print the residue information. Okay. So if you execute that, you'll see the neighbors. We see that the neighbor of this is histidine 93. So our analysis shows that histidine 93 is a neighbor to the iron in the heme group. And so the protein structure that we're analyzing is pretty well known. This is a known thing that the heme group binds to histidine in myoglobin. And this histidine is something called the distal histidine, and it's known to bind to the heme group. You can read more, if you're interested in reading more about myoglobin structure, I've included a link here that gives you a really detailed breakdown. And so now what I want you to do, so go ahead and someone try increasing this distance to 5 and performing the same analysis. I'll actually demonstrate that. So now we can see the nice thing about this type of analysis. So let's say we want to see what the maximum distance for a neighbor is, but we want our maximum distance to be 5. So I'm just going to go here and change my cutoff distance, and then I can run the same analysis. And then if I go down here, now I'm going to see histidine 93 and histidine 97, which if you read about myoglobin structure, there's something called the distal histidine. And then I think it's like the proximal histidine. That's what this other one is, I think, or maybe it's 64. I'm not sure which histidine it is, but you'll just see that we see a lot of histidine residues here. So it allows us to kind of easily change the distance that we're analyzing. Okay. All right. Any questions about this part? Okay. So we are seeing histidine 93 appear 3 times because we're printing information for every atom that is a neighbor. So we're looping through all of the neighbor atoms, and then we're printing the element and the residue name. So what that means is that there are 3 atoms in this residue that are within 5 angstroms of the iron atom. And you can see that it's a carbon atom, and a nitrogen atom. And the residue numbering does correspond to the numbering that is in the crystal structure. So it probably starts from 1, yes.
Yeah, and that's N terminus to C terminus as is the convention.
So we had a question, I'll answer this. This isn't quite about what we did. It's a question about whether all of the structures in the PDB database are experimentally determined. So all of the ones that have PDB IDs that look like this are experimentally determined. So if you look up here, you'll see that there are 203,000. These are experimentally determined structures. And then this is new. I'm not quite sure how long it's been around, but now there are structures that are not experimentally determined, but are computed structures. And there are 1 million computed structure models. So they call those CSMs. If you want to, usually these don't show up by default, but if you want them, you can indicate in the search that you want those. Okay.
And if you look at those lists, the list you just had there, Jessica, on the PDB website, notice that there's a little flask in black next to the experimentally determined structures, and then there's a blue computer terminal next to the one for computed structure models. I think they did this last October, and so that's how you can tell when you get a list up of if you do a search and include both, you can tell which ones are experimental because they have the black flask next to them, and the ones that are computed have the blue computer screen.
Okay. And then we had a question about why we have a 1 here. That's just because this is a collection. And we so we had a question on why this 1 is here. So if you look at what we're printing differently, it's just because I just wanted this number. This is something that's similar in Python to a list, but it's like a tuple, but I just wanted the number instead. So this would be index 0, this would be index 1, and this would be index 2. I just wanted the number, so I'm using this 1 here to just get that number. Okay. Great, good questions. Okay, so I do want to keep answering questions, but we will have, we're gonna kind of reconvene at 4 o'clock, but we do have an exercise here for you to do. Actually, why don't we we'll give you until 5 minutes after the hour, and you can either take a break or you can try to do this challenge. So, and I think that I might have made a mistake. If you pulled the repo last night, you might not have this image, but I'll keep this here for you to look at. So a challenge for you is to repeat this analysis for something called a zinc finger structure. And they're small protein motifs that are characterized by zinc ions. So this is an image showing the crystal structure I'm going to ask you to analyze, which is called A1, 1A1T, and it's the structure for the HIV-1 nucleocapsid protein, which contains two zinc fingers or two zinc ions. So my challenge to you is to see if you can use what you just learned to figure out what the neighboring residues for this zinc finger or for these zinc ions are.